In this video, we're going to talk about neoplasia. Specifically, we're going to address cancer prevalence and the behavior of neoplastic cells. Or perhaps we should say the misbehavior of neoplastic cells. I like to start any discussion with information that can help you see why nurses and health professionals need to know this particular information. This data comes from the American Cancer Society, and this slide shows us trends in death rates for women in the United States from cancer between the years of 1930 and 2017. If we start on the left hand of the slide, notice that I've underlined stomach cancers, and that's that dark black line. And the slide depicts that stomach cancers have been, deaths from stomach cancer have been decreasing over the past several years and are really very, very small at this point in time. This is probably related to the development of drugs that help manage stomach acidity and to control a specific bacteria that irritates the stomach and can cause cancer, and that's called H. pylori. We are going to talk more about H. pylori in the GI section of this course. You can see the cancer with the highest rates of death is lung cancer. And lung cancer remains the top cancer killer. And notice how it peaked around 2000 and is now trending downward. Colon and rectal cancers are also trending downward. Likely this is due to better screening and earlier diagnosis, since people are getting colonoscopies every 5 years or so once you become of age. And breast cancer is also on a downward trend. And I would attribute some of the success with breast cancer to better therapies, more knowledge about the genetic influences associated with breast cancer, and also better screening for early detection. This slide depicts cancer death rate trends in men. Note that the cancer rates for lung and bronchus are nearly twice as high for men as women. This is likely attributed to the overall higher smoking rates in men. Stomach cancers have also been significantly reduced. They started out significantly higher than for women. Colorectal cancers are the light blue line, and again, they have trended down since about 1985. Notice that prostate cancers had a bump up in 1995. This might be due to better screening and improved diagnostic markers. And we'll talk more about diagnostic markers later in this chapter. So don't memorize anything from these two slides. Again, they're just here to highlight the importance of understanding how cancer develops in response to irritating stimuli and how new developments in diagnosis and treatment can make a difference. What is neoplasia? When you break down the word neoplasia, notice that neo means new, and we know plasia refers to growth in terms of cellular multiplication. So neoplasia is new, uncontrolled, uncoordinated cell growth. It is cell replication that is not under physiologic control. It's important to know that neoplasia is not synonymous with cancer. Although you may hear the words used interchangeably, a neoplasm refers to a tumor, and tumors may be either benign or malignant. Before we talk about features of abnormal cells, let's review the way that cells should behave. I'm sure you learned about the cell cycle in physiology. It's a 5-phase cycle that begins with the GO phase, where the cell may leave or re-enter the cycle. In G1, the cell begins increasing the number of intracellular structures in preparation to divide. The S phase marks the point where DNA is synthesized, and in the G2 phase, the cell will replicate its RNA and other proteins. And finally, the M phase is the point at which the cell divides. With normal cells, the number of cells that die from apoptosis is equal to the number of cells that are produced. This keeps the number of cells in our body at about the same at any given time. Cancer cells are multiplying way more quickly because fewer enter the G0 phase of the cell cycle. They continue to divide until limitations in blood supply or nutrition limit their growth. The growth fraction is basically the rate of mitosis. It's the ratio of dividing cells compared to the number of resting cells. And this explains how tumors can grow in size relatively quickly. The doubling time is just as it sounds, the time it takes for a tumor to double in size. And tumor growth rates are widely used for prognostic purposes. Yes, we can expect cells to behave and replicate in organized and predictable ways, but not every type of cell follows a similar life cycle. In fact, depending on the type of tissue, the ability to proliferate will vary. Labile tissues are those that are continuously dividing, like skin and epithelial linings in the GI tract or in the respiratory tract or vaginal and cervical linings. These cells are constantly being sloughed off, and it makes sense that we kind of need them to continuously divide in order to replace the cells that we've lost. There are some type of cells that divide during normal growth and development, but once growing stops, they tend to stop dividing. These are called stable tissues. They don't enter the cell cycle unless something happens to stimulate regeneration. For example, if you donate a lobe of your liver, liver cells are capable of regenerating until your liver is back to its previous size. But liver cells won't begin mitosis until something happens to create that deficit in liver tissue. Finally, we have permanent tissues, nerve cells, skeletal muscle cells, cardiac muscle cells, and these are not capable of mitotic division. This has a lot of clinical significance for heart patients in particular, because once someone has a heart attack, the muscle that's lost will never be regenerated. Growth factors are key to the discussion of proliferation. Without growth factors, cells can't proliferate. However, a special feature of cancer cells includes the fact that they are able to replicate without growth factors. Cells only divide when growth factors tell them to do so. Growth factors stimulate stable cells to enter that cell cycle and divide. However, cancer cells behave quite differently. They are able to divide without growth factors from elsewhere. Some of them make their own, while others produce lots of active receptors that will help them replicate. In fact, some antibody-type drugs have been developed to help interrupt cell division by blocking the extra receptors on the cancer cells. Cancer cells behave more primitively in that they only keep the functions that they need to survive and to proliferate. Although our immune system should be able to recognize and get rid of abnormal cells, it is thought that by producing oncofetal antigens, the cancer cells are able to go undetected. Differentiation refers to a cell's ability to become what it's supposed to be. We expect immature cells to develop into a specific type of specialized cell that has the ability to perform as that type of cell. For example, we expect an immature esophageal cell to grow into a fully functioning esophagus cell and be able to do its job within the GI system. However, if cell replication goes awry and these esophageal cells are multiplying in an uncoordinated way, as in neoplasia, then they can become stuck in one phase of differentiation, and they never become able to do their job. In the case of esophageal cells, this would be to contract and to move food from the mouth to the stomach. But when cells become stuck and began to multiply in a more immature phase of differentiation, then we say they are poorly differentiated. And when you look at poorly differentiated cells under the microscope, it may be impossible to even tell the type of cell that they started out as. Benign neoplasms tend to be well differentiated, meaning they tend to resemble the normal cell, but they are not able to control proliferation. In contrast, malignant cells have lost the ability to control both proliferation and differentiation. This is cancer. So remember these features of cancer cells. They can't do the job they were intended to do. They grow in the absence of growth factors, and they evade apoptosis. Here are two more properties of cancer cells that you'll want to remember. The first is contact inhibition. Imagine that you want to enter a room, but it's really crowded. You would have to squeeze in. So you would probably wait outside the door until a few people left so that there was enough room for everyone. Cancer cells, however, are very rude. They would enter the room and they would continue to multiply. Being crowded does not stop replication as it would for a normal cell. Normal cells are also way more social than cancer cells. They like to be connected to other cells or anchored to them, or to be anchored to another surface. If they're not anchored, then they will stop dividing or even die. Cancer cells don't need to be anchored to anyone or any other cell. They still divide and grow. Cancer cells tend to be unstable genetically. They're not able to correct errors in cell division, even serious errors like multiple types of chromosomes. These gene mutations affect the ability of the cell to regulate growth, to enter the cell cycle, and to stop multiplying. Cancer-associated genes that are involved with growth promotion include proto-oncogenes and tumor suppressor genes. Proto-oncogenes communicate normal cell division to the cell proteins, including growth factors, growth factor receptors, and cell cycle proteins, and apoptosis inhibitors. When proto-oncogenes mutate to oncogenes, the function is activated or increased. The mutated gene causes errors in cell formation and replication. You may recall that RAS proteins are responsible for turning on genes involved in cell growth. Overactive RAS signaling can lead to cancer. The Philadelphia chromosome, you're going to learn in this course, is a genetic defect associated with a specific form of leukemia. In breast cancers, you've probably heard of the BRCA gene, but also tumors are checked for HER2/neu gene. The HER2 is a protein that promotes the growth of cancer cells. HER2 positive breast cancer tends to be more aggressive compared to the other types, but the prognosis for HER2 positive cancer is actually quite good. Tumor suppressor genes inhibit cell division. Mutations in tumor suppressor genes means that the genes are no longer able to help stop the replication of cells. So defects in either proto-oncogenes or tumor suppressor genes may lead to cancer. This slide also depicts a few more examples of genetic mutations that result in oncogenesis or cancer development. We talked about the mutated RAS gene on the last slide. We also mentioned the HER2/neu, and the p53 is a gene that normally makes a protein that helps to control the proliferation of damaged cells. So without functional p52, genetically damaged cells will continue to replicate. Also, there is a gene that helps in regulating apoptosis or normal cell death, and that's the BCL2. Most normal cells will die eventually. However, cancer cells are considered immortal. Remember the telomeres from physiology? These are the tips of our chromosomes. With every cell division, most cells will lose more of the tips of the chromosome or the telomere. The enzyme telomerase helps to replace the shortened telomere. Cancer cells have the ability to turn on a gene that promotes more telomerase. And more telomerase means longer telomeres, and thus cancer cells are considered immortal. In review, this table will summarize the important differences between benign and malignant neoplasms. So it would be good for you to know all of these features and the other highlights from this slideshow. In part 2 of chapter 6, we're going to continue our discussion of cancer, but not so much in terms of the cellular behavior. Rather, we'll talk about how tumors are named, how they invade and metastasize, how tumors are diagnosed, including grading and staging. Although cancer treatment is quite complicated and it certainly varies for each type of cancer, we're going to take a look at how cancer is treated at a very high level.